Okay, so this is a special part of our program, and on behalf of Cold Spring Harbor and the Germ Cells meeting, I'd like to welcome representatives of the Gruber family and the Gruber Foundation here to the meeting, and welcome all of you to the 22nd annual awarding of the Gruber Prize in Genetics. And the organizers and David Stewart, who runs conferences here at Cold Spring Harbor, we were all very pleased when the Gruber Foundation reached out to us to have the award ceremony here at this meeting, because this year's awardees are very important contributors to our germ cell community and people that have meant a great deal to us and contributed a great deal to our understanding of germ cell biology. So this is really wonderful to have it here and extremely appropriate. And I would just like to say on a personal note that I really appreciate and think it's wonderful that the Gruber family has elected to use some of their resources to support science and to recognize scientists, which is of course particularly important during a time that we feel that at least some of our country and maybe other countries too some people are just not relying on science the way they should be for our national dialogue and for decision making and for policies. And so I'm going to turn things over to Sarah Rea, who is the executive director of the Gruber Foundation, who's going to then run our program for tonight. So please, Sarah.
Thank you, Professor Van Doren. We are delighted to be here at Cold Spring Harbor Laboratory to present the Genetics Prize at the Germ Cells meeting. While we are here to honor Ruth Lehmann, James Priess, and Geraldine Seydoux, let me mention that they join Cosmology Prize recipient Frank Eisenhower and Neuroscience Prize recipients Larry Abbott, Emery Neal Brown, Terrence Sejnowski, and Chaim Somplinski on our 2022 roster. Please note that nominations to the 2023 Gruber Prizes are open until December 15th, 2022, and that we encourage nominations that reflect the breadth of the fields and the diversity of those working within them. Before we return to genetics, I simply must acknowledge our co-founders, Peter and Patricia Gruber, whose combined vision and leadership established the International Prize Program, and whose care in doing so gave it the legs to stand on its own. And let me now invite Jane Hubbard to say just a few words about our collaboration with the Genetic Society of America.
Thank you, Sarah. So speaking as the current president of the GSA, as many of you know, the GSA, despite its name, is an international organization that supports the genetics community with conferences, publications, science communication, equity work, and more. Current and past GSA board members and presidents are here in our audience. The germ cell community has been a big part of the GSA for a long time. So for those of you who are new to the field, I encourage you to get involved. It's on behalf of the entire GSA community that I want to express how pleased GSA is to collaborate with the Gruber Foundation for the Genetics Prize. GSA also collaborates with the Gruber Foundation to administer the Rosalind Franklin Young Investigator Award, which Gruber funds. This award honors groundbreaking contributions of Dr. Rosalind Franklin. It honors her contributions and her inspiration by supporting new generations of women scientists in the field of genetics. And so with that, I will turn it back to Sarah. Thank you.
Thank you, Professor Hubbard. Our relationship with GSA is invaluable, and we appreciate you joining us here today for this. Ruth Lehmann, James Priess, and Geraldine Seydoux join an illustrious laureate list. They were chosen by a distinguished selection advisory board nominated to us by GSA. We sincerely appreciate the knowledge, commitment, and enthusiasm that these advisors bring to the judging process. I would like to now invite Alan Spradling, the chair of this board, to present the citation to the prize and say a few words about our recipient's scientific accomplishments.
Well, it's truly a pleasure to be in the position of presenting this award today, because all three of these winners this year have personally been very inspiring for my own research, and I know that for many of you, that has been the same. So allow me to start by reading the citation. It says, the Gruber Foundation proudly presents the 2022 Genetics Prize to Ruth Lehmann, James Priess, and Geraldine Seydoux for discovering how early nematode, Priess and Seydoux, and fruit fly, Lehmann, embryos set aside particular cells during development, especially germ cells that result in intergenerational inheritance. Their research employed genetic screens and elegant molecular analyses to characterize mysterious germ granules located in oocytes and early embryos. Germ granules house mRNAs whose regulated translation and localization specify germ cells and protect them from premature transcription. Germ granules utilize a novel mechanism, phase separation, relevant to many other particulate cellular structures and to organismal development. These seminal genetic findings illuminated normal biology and furthered our understanding of nuclear inheritance while highlighting the power of model organisms to reveal the mysteries of life. So, that is, truer words were never spoken, but I would also just like to add that these three individuals, as I have just been so inspiring to the enterprise of developmental genetics, and particularly its application to germ cells. After seeing how Ruth Lehmann could do a genetic screen in a field where no one had ever found, a really useful mutant, and so. And suddenly we had a whole pathway of germ cell specification. And James Priess, did fabulous screens that found wonderful genes in nematodes, such as Pi1. This was very inspiring. And I also, the way that Priess analyzed nematode development, showing for the first time that there were important cell-cell interactions and signal transduction pathways between nematode cells that guided their differentiation. It was really a revelation, and that the reason that classical embryologists had thought there were two types of embryos, mosaic and regulative, and you might have had to learn two different whole systems of development. But it just turned out that in the simple system of the nematode with relatively few cells, the same processes and signals, just gave you a very reproducible result. So it seemed to have a system where you would get an invariant lineage. And this, of course, means that whatever model that we study, it's relevant to all the other animals, including humans. And we've seen so many examples of that in the meeting. And then Geraldine, first with her work on the polarity genes, the PAR genes, which were discovered in the Priess lab, and then later with her incredible analysis of the germ granules, just showed that we really need to look at the cells that we're studying and to see their cell biology. And that is, the basis on which the developmental biology can be understood. So I couldn't think of a more appropriate location in which to present this prize at this germ cell meeting. And I would like to now ask all three of our awardees to come forward and we'll present the prize.
Thank you. So needless to say, I'm thrilled, and I hope I can go through that 1 minute of thank you. This is really, truly a wonderful honor, and I am so grateful to the Gruber Foundation, to the Genetics Prize Advisory Board, and to the GSA for honoring our work and celebrating genetics, which is a discipline that we all know and love. I also would like to thank my lab members, past and present. Subu, where are you? Want to see you there. And my other graduate students who are here today. This has been a wonderful ride, and I'm so glad that you were able to share it with me. I also would like to thank my co-recipients. These guys definitely led the way, and I am so grateful to being able to walk into their footsteps. And then finally, I'd like to thank all of you here. This is the meeting that I've been going back to since that famous Colorado meeting. And as they say, it takes a village, and this is a really good village to belong to. Thank you.
Well, I'd like to thank the Gruber Foundation as well. This is quite an honor for me, and I think it speaks highly of the work that many, many people did in my lab, some of which I'll mention in the brief talk I'm going to give. I'd also have to thank my wife, June, for all her support. And,
as was already expressed so well, I really want to thank the Gruber Foundation. I think it is so important that basic science, fundamental knowledge is supported, and genetics is sort of the method and the question at the same time, the method which allows us to make unbiased discoveries, and that is at the core of fundamental research. And so we are grateful because with an important prize like that, winning an important prize like that, we can also be spokespeople together with GSA to really send the message that we do need fundamental research, and we thank the Gruber Foundation. I also really thank the Gruber Foundation for the support to the Rosalind Franklin Prize. I think this is so important to support young faculty and to support women in science, and so I'm very grateful. It means a lot to me to have gotten this prize. And at the end of my talk, I'm going to thank my lab. Thank you. Okay, so I want to make a few, so each of us will present something, and to be honest, as Geraldine said, you know, we are here sort of in family in a way. We know each other for a long time, so it's kind of difficult to say what do you actually present here, so I'm going to try something. I'm calling this a germline life cycle, so it's sort of also my life cycle, but that's at least how I feel about it. And so reproduction is enormously important. Reproduction is actually the cycle of rejuvenation. It's the cycle of how we go from one generation to the other. And it occurs in every organism. It has to occur in every organism when sperm and egg meet, and then a fertilized egg cell will develop into an embryo. And at some point, primordial germ cells have to be set aside. And they're not necessary for doing anything really to the organism and the organismal survival. They're really important to get to the next generation. And there are many questions that we need to understand this very, very specific and specialized cell type, starting with specification and ending with meiosis and the differentiation into egg and sperm. So like to sort of make a sort of a thought experiment, and that is if we think about this idea of the hourglass of development, which many of us developmental biologists are very well aware of, and that is that early stages are not very similar, even if we're looking at vertebrates here. But there is this particular stage in development, and this is just when segmentation occurs, where sort of all organisms look actually very, very similar, and Haeckel had already pointed this out several centuries ago. And then later, the organism looks very, very different. And it was during actually the end of my graduate work that it became clear that this wasn't just a principle of vertebrates, but that this principle could actually be extended, and that we could include flies and worms, and we all had very similar genetic makeups, and we could learn from each other even when we're working on very different organisms, which is so clear, I think, at this meeting. I think every germ cell meeting I come to, it seems to be clearer and clearer how much exchange there are and how much we're working on similar questions. And so the phylotypic stage in the fly embryo would also be the stage where the homeobox genes the Hox genes are aligned in their specific pattern. But then the egg is not a phylotypic stage. The egg is really different. The egg is laid at a very different developmental stage. And I think that is really important to not stress about, how similar is this egg to that egg? Because the eggs are all very different. And just to put one example, a human egg basically has zero information when it's laid, and it has to do it all on its own. It even has to make its own extraembryonic tissues, while the fly embryo is completely patterned. It knows everything. It knows where the head goes, where the tail goes, where front and back goes. And so it isn't surprising that when we're looking at early stages of development, that we see big differences, because it isn't a phylotypic stage. And so there are idiosyncrasies. So while there are similarities, there are also idiosyncrasies. And so one idiosyncrasy of the Drosophila embryo is that germplasm that you have heard about a lot in the last couple of days. And the RNAs are localized, and these RNAs segregate into germ cells during the early stages of development, and they're patterned within the germ cells, and the middle germ cells actually get more RNA than the ones surrounding, and they're going to be more successful than the other ones. And so this is already what the mother had put into the egg, already tells the embryo which cells are more successful in their migration towards immortality. And so what's even weirder and more idiosyncratic of the germline is how actually the whole early embryo in flies forms. And that is that there are two completely genetically independent cellular mechanisms of making cells in the early embryo. One mechanism is and this is all because there are these nuclei which divide. There are no cell divisions early on, so completely different to most of the organisms that non-fly people work with. And cellularization is formed by ingrowth of cells, and that makes all the somatic cells. It's an odd way because there's actually a polarized ingrowth, and it absolutely requires zygotic transcription. So the embryo has to be genetically active. Its genome has to be active. Then there's this very, very different pathway which makes the germ cells, which form before the somatic cells form, and completely triggered by maternal transcripts. And what is happening here is it's even a completely novel type of cellularization, where two orthogonal constrictions generate two cells. So one is the normal anaphase constriction, and the other one is this bud constriction, which is spindle independent. And that leads to the formation of germ cells in a totally maternally driven process. And what's interesting is how this happens, and it's not trivial how this happens, because the determinant gene here, called germ cell S, which is one of those localized RNAs, encodes a ubiquitin ligase adapter. And that ubiquitin ligase adapter, its sole function seems to be to degrade torso, which is a signaling molecule that's important for the front and the back of the embryo to pattern. So you don't want to use patterning genes, somatic patterning genes being active in the germ cells. And so indeed we know this is a sole factor, because if we remove germ cell S and torso, germ cell formation is restored. And so what germ cellus does is it keeps the signaling molecules which are activated by torso receptor tyrosine kinase at the front and the back away from the germ cells so that the germ cells can form. There are other factors, other RNAs which are localized to the germ plasm that are important, like nanos. And germ cellus, nanos, which is a translational repression molecule, which by translational repression suppresses the soma. I already told you about germ cellus as a E3 ubiquitin ligase, degrades the somatic pathway. And then finally, polar granule component, which is a PTFB competitor, which represses the zygotic transcription and thereby makes sure that transcription only occurs in the soma. Here is actually a torso target, which you can see is absent, is not expressed in the germ cells, and the germ cells do not express active POL2. So why I started off saying these are idiosyncrasies, because there are RNAs which are localized maternally, and in many eggs, RNAs are not localized and specifically important for germ cell fate. What is common is the principles: degradation of somatic signals, translational repression of somatic signals, and repression of zygotic and somatic transcription. So those are the general principles, and those are important. And whether they are produced by maternally localized RNAs or transcribed in the germ cells themselves, the output is the same. And what we found through the years is that the molecules are the same, which is, of course, best displayed by nanos being pretty much a unique factor which is found in all germ cells. So I want to turn now to tell you a little bit about science. This was my philosophical part. Now I'm going to come to tell you a little bit about science, and I want to tell you about something which is not idiosyncratic, and that is germ cell migration, because pretty much all germ cells migrate. Sorry to the worms. They turn it around. The endoderm comes to the germ cells. But otherwise, there's pretty much in every organism. And is this still a curiosity? Is it because the germ cells are formed sort of outside? Because of this not having to respond to somatic signals? Or why do they have to migrate? But it is a fact. And so in that regard, for germ cell development, this is certainly one of the phylotypic stages, and we could probably think of others. And so I want to tell you a little bit about what we've learned about germ cell migration in the last couple of years. And so shared by all organisms is that in most cases, except for the zebrafish, germ cells are formed in one clutch at some location, whether it is by maternal determinants or by induction between tissues. They then move during morphogenetic movements. They often undergo transepithelial migration. Since they're in a clutch and you have two gonads, they have to separate at some point into two groups, and then they directionally migrate to the gonad, and then absolutely require the somatic cells, which we usually sort of put down, but they are absolutely critical for normal gonad development and the differentiation of the egg and sperm. So this process occurs in flies, it occurs in fish, it occurs in mice, it occurs in humans, and just takes different time, but germ cells always do this migration to meet up with the somatic part of the gonad. And so we have used, and this is one of the parts where we have used genetic screens to identify the genes which are involved in germ cell migration. And I remember everybody's talking about this Colorado meeting. I remember at the Colorado meeting where people told me, oh, you should just look at integrins and this and that, what other people have found, what's important for migration. And I said, no, that's not my approach. I already knew that integrins weren't working for my postdoc. But the approach was to just have an unbiased screen. Let's see what we find. And the power of the unbiased screen is that sometimes you find, you go by the phenotype. You go by what has the most interesting phenotype, what is the strongest phenotype. And sometimes you kind of have to suck it up that maybe that's not the gene that you really wanted, right, Mark? So we'll get to that. So I'm going to tell you first how germ cells actually migrate. And this is the work of a postdoc in the lab, Ben Lin. And so this is at the very beginning when the germ cells, they form at the posterior pole, and then through these morphogenetic movements, they get inside the embryo, they're internalized, and now they have to move through the posterior midgut as the posterior midgut loosens up. So they're taking advantage of the loosening of the posterior midgut to move out prior to sorting into the two groups. And here you can see a beautiful movie of watching this migration. The early stages of migration are very, very hard to follow because they're inside the embryo and it's full of yolk, and it was only the employment of two-photon microscopy which allowed Ben to really study the process. And so here is Ben, and Ben really brought live observation to this difficult question. And it is very interesting, once we started to observe and study germ cell migration only live, we had to correct and revise some of the conclusions we had drawn before, because it is the dynamics that is so important to figure out how cells migrate, obviously. And so what Ben observed very early on is that the germ cells seem to never change their shape. They don't seem to have an extended leading edge, and they have this very extreme concentration of actin at the lagging edge. And what he found then was that they're not, rather than being migrating by actin protrusion specifically at the leading edge, actin flows through the germ cells globally along the cortex. And he sees this in vivo, and then he takes the germ cells out, and he can measure this more precisely, where he can show that the F-actin flow and myosin go together to give the propulsion and the directionality. And so we think that the flow of actin and myosin leads to the directionality by creating a lagging edge concentration of myosin. And so then he wanted to understand how is this actin polarity organized. Obviously we know quite a bit from, again, other systems, and most systems for cell migration are either initially they were on culture dishes with 2D, and now they're more in more gel-like conditions. And there are very clear ideas of how cells migrate. And so in the front, it's known that RAC and CDC42 can propel the branched actin, which then leads to these extensions at the leading edge, while at the lagging edge, it is often Rho A, which courses myosin for the contractile forces and can also get the linear polymerization of actin. So we already knew from previous studies that RAC and CDC42 were actually not required in germ cells, and in live imaging, Ben showed that clearly these were not playing a role, they were not needed. So this left us with really explaining how the germ cells can propel themselves by this global actin flow plus the back pushing. And so what he focused on and what he realized with Rho A sensors, that this was the power of this of the action of concentrating the myosin in the lagging edge was due to Rho A activation. And so he did a genetic screen to try to figure out which ROGEF may be involved in this. And in the screen, he identified ROGEF2. And ROGEF2 was also beautifully localized exactly at this lagging edge, just before the germ cells explode when they go through the gut. So at that point, they're in this kind of rosette shape before they individualize. And he then looked at what was the function of ROGEF, and what he could do is artificially he could activate or degrade ROGEF in the germ cells specifically. And you have to think about, this is not trivial because this is all maternally driven. And so what he has to do is to maternally deplete, put back in the soma and then study the germ cells themselves, which are only depleted. Because, of course, if the soma is defective, we can't really make a conclusion about how the germ cell migration itself. And so what he found in terms of the global actin flow, that it was definitely reduced when there was degradation of the ROGEF2, and that it was increased when there was activation of ROGEF2. And so it seems that ROGEF activity levels are a dial of how much actin flow there is. And in both cases, germ cell migration defects will be there. So dialing it just right is very important. He next looked at, so this gives you just motility. Where does the directionality come from? And we knew that the directionality for germ cell migration comes from a G protein coupled receptor called TRE1, but we didn't really know what the signaling pathway downstream was. And so he looked then at the ROGEF concentration in TRE1 mutants, and what he found was that there was clearly reduced polarization and reduced intensity of the signal, suggesting that TRE1 was mediating the orientation of the migration. And so to summarize many experiments, so I already told you that that ROGEF is required for the flow speed, and this is through the traditional, what one would think of signaling pathways of a ROGEF, which means activation of Rho and also its localization to the membrane. But there was, when we were thinking about how the motility is coming about, it was, again, a discovery from genetics, and that is actually asking about mutants, and in this time it is mutants which are specific to the specific regions of the protein. Mutating the G-alpha-12-13 binding site, which was actually in other instances, is important for directionality, is important for ROGEF to be released from microtubules, had absolutely no effect on germ cell migration. And we already knew that from mutating G-alpha-12-13 itself, that it didn't affect germ cell migration. So he was left with this domain at the N-terminus, and he realized through sequence analysis that this region was, on the one hand, able to bind microtubules. It had an EB1 binding site, but it also was regulated by AMP kinase. And so he then discovered that there was a completely different mechanism downstream of migratory GPCR, which uses AMP kinase to release, through phosphorylation, the ROGEF to then provide the differential migration. And so what I want to kind of leave you with this vignette is that germ cells do not migrate, and this is not so different from what Raz in his detailed analysis of germ cell migration in the zebrafish has found, but rather by a cortical actin flow that is then directed by the GPCR. And in this case, ROGEF2 is kind of the master regulator through its multimodality to create both the actin flow plus the directionality. So now I'm going to come to the second part of germ cell migration, and that is how to settle down in the, into the gonad. And here, and this is why I was pointing especially to Mark. So many years ago, Mark, in a genetic forward screen, he identified a gene which he thought, what a bummer, how am I going to ever get a job with finding HMG CoA reductase? It worked out well, because here is a housekeeping gene, and now, we may not find that so surprising anymore, but at the time, that a housekeeping gene was specifically expressed in the somatic gonad, and that it had actually a sort of master regulatory way of behaving, because if you knock it out in the somatic gonad, germ cells will be lost. But if you express it highly at a new location, it can move, germ cells can move to that new location. So it was both necessary and sufficient for germ cell attraction. But it's a really it's at the beginning of a big pathway. So how does it work? Clearly it's dose sensitive for something. And for a long time, that remained unclear until Lacey Barton came to the lab, and she looked at the entire pathway, which we had started to dissect, and her eyes fell on juvenile hormone. She thought perhaps, and with a bunch of kind of preliminary data, she said maybe juvenile hormone is the mediator. Because what she saw was that a juvenile hormone synthesis enzyme was expressed specifically again in the gonadal mesoderm, just where the germ cells are coming to. And she could show purifying germ cells and just giving them juvenile hormone was enough to attract these germ cells through a Boyden chamber assay, which we had previously shown that some activity was in conditioned medium, and clearly aspects of that is juvenile hormone. So how does juvenile hormone attract the germ cells? So we know now that HMG coreductase works through the juvenile hormone synthesizing enzymes and then somehow attracts the germ cells. So with juvenile hormone, it was pretty clear what one would ask, and that is there is a juvenile hormone receptor which then acts in the nucleus to activate transcription. So she made reporter fusions where she uses the promoter elements to activate the juvenile hormone receptor mediated GFP, and you can see here how nicely that works, but you may already notice that the germ cells are not expressing this reporter while the reporter works nicely and is dependent on the receptor, which is produced by MET and GCE, and while in the somatic signals are largely gone, the germ cells don't express this receptor, suggesting is it perhaps not through this receptor? And so again, she asked whether perhaps transcription was not required for the attraction, and so she both mutated the receptor, but also in this migration assay, just in vitro, she just blocked transcription. And it's very clear, even the KC conditioned medium doesn't care whether there's transcription. So again, the germ cells are not transcriptionally, they're not even very active at that point transcriptionally, and they don't respond with a transcriptional response to this guidance cue. And the same is when she's given juvenile hormone. And so she has very good evidence that there is a GPCR which receives this signal, and this is then through a calcium cascade that then promotes the migration. And she will be following this up in her own lab now at UT San Antonio. But Lacey also wondered, juvenile hormone looks very much like retinoids. There are very many similarities. And it is also that the retinoic acid synthesizing enzymes at the time when the germ cells in the mouse move towards the gonad are expressed in both sexes. They're later sex-specifically expressed. And so she wondered whether retinoic acid could have the same role in guiding germ cells to the gonad. And so first she asked if retinoic acid in the in vitro assay could attract germ cells, and indeed it can. And then she asked whether mouse germ cells could be attracted. So first she had to develop this same assay for the mouse, and here's a nice control. We know that STF1 through the CXCR4 receptor attracts germ cells, and this is working in this assay. And indeed, retinoic acid can attract germ celys. And we did this in collaboration with David Page's lab, and then did most of the work at NYU in the Skirball Institute. So to summarize, lessons from the fly, which have some relevance for germ cell migration, and that is that germ cells migrate, and I think every migratory cell has to be looked at for its own mechanism. We shouldn't just take a textbook idea of how cells migrate. And so the classical protrusion-based mechanism is not a case for fly germ cells, and it may not be for other germ cells. ROGEF integrates basal global cortical actin flow with directed migration. Importantly, in terms of directly going between the fly and the mouse, juvenile hormone or retinoic acid are drivers of germ cell migration. And obviously, we want to learn much more about this. And so the two heroes of these stories is Ben Lin, who is still in New York and is on the job market, and probably many of you got his application, so look at it carefully. And Lacey Barton, who just left and is setting up her own lab now at San Antonio, and we had many collaborators, and we had good funding there. And now we move to MIT and the Whitehead Institute, and this is our new group. And you can see we Tony Mahowald has joined our group, so now I'm the boss of the boss of the boss of the boss of the boss. But we also see ourselves now as a really wonderful germline community. And finally, I want to thank my lab now, and these are all my graduate students on the left, all the postdocs. Oops, no, I can't go back. Oh, that's sad. Oh, no, maybe I can go back. Yeah, here we go. My graduate students on the left, postdocs on the right, and some of you are here. Actually, some of the postdocs who are here and are the students from former postdocs are here, and this is just, there can't be anything better. And it's always a little sad that these prizes are given to a single person because you are so much smarter than I am, and so I'm just using you to get these great results and be entertained and have fun and have people who I can have a great discussion with. And so I am just so happy to have been able to have such a wonderful group, and I also to be part of such a wonderful community. And so with that, I thank you all very, very much.
Okay, so let's see. Okay, from an early age, I've been interested in embryogenesis, like many people, and as an undergraduate in graduate school, there was you can call it a great divide in how people thought about embryonic development between the basic phenomenon is that if you take the fertilized egg, and it normally becomes two different types of cells, in one type of animal you could split these apart and then regenerate a whole animal. But it's been known for over 100 years that in nematodes something else was going on. And this began with the finding in ovary that there was actually a loss of chromosome material from one of these cells, which was from the somatic cells, but the chromosomes were maintained in the cell that became the germline. And then there was a very nice study, again in parasitic nematodes in 1909, where Stevens took these synchronous nematode embryos, put a quartz cover slip on top of them, and then laced fragments of aluminum foil, and then shone a UV light on them. So it was like an early idea for a microbeam. And then he would look at the dead cells and the embryos, and what he was seeing is that it looked like that various early cells were becoming very, very different things. And so the basic idea is in these types of embryos that if you kill a cell, you can't, the A will make A but not B, and that if you separate the cells, one makes A and one B. And back then, in the absence of molecules, there was a lot of emphasis on nomenclature. And so this was called indeterminate, regulative, cell-cell interactions. And Sidney Brenner coined the term American plan, where environment determines fate. And here, these were called determinative, mosaic, cell-autonomous, European plan, where ancestry determines fate. Then in the modern era of C. elegans, there were work continued to show that there was early differences between the blastomeres and some of the inspirational papers for me were these, and particularly Susan Strom's work, where she found an antibody that would stain pea granules, and it showed these granules were localized specifically to the germ line. And there were the other experiments with cleavage block experiments, et cetera, that suggested these somatic cells were becoming different from each other. Now, as we began to understand more about the lineage, it became clear that there were some of these early cells were making clones of tissues. But then the analysis of the lineage went into full steam with John Solston, who decoded the entire cell lineage. He was my postdoc advisor, and this is sorry. Anyway, excuse me. Anyway, sorry. Okay. This is a lot of work. Here, and his conclusions were that the lineage was highly complex. Highly complex with relatively few examples of clonal cell origins. And the remarkable feature was that it was essentially a variant between different animals. This is an important point because what you're looking at is in these early cells, what he was seeing is that they underwent a specific lineage. So these lineage diagrams represent anterior, posterior cleavages. And so what he was seeing in general is that these precursor cells weren't just producing a random collection of differentiated offspring, but rather specific cell types in a specific anterior-posterior order. And his conclusion was that the cell lineage itself, complex as it is, plays an important role in determining cell fate. And he did a large number of experiments after the 50 cell stage, and he saw no significant cell replacement. Okay, so from work in Drosophila, it was becoming clear that the way to approach this study or of how these early cells become different is to do genetics. And so work in the 1980s and particularly the discovery of the maternal gene bicoid in Drosophila really was clear that that was the way to go. And so there were some small-scale maternal effect lethal screens in C. elegans, but it occurred to me that to find out what we really wanted was to look for informative genes and to do a much larger screens. So the basic idea, well, it occurred to me that C. elegans was really an ideal system to look for maternal effect lethals because you could take advantage of two things. It's a self-fertilizing hermaphrodite, and there were also mutations. Oh, thank you. I can't really cry. That's okay. Okay, so there are also mutations that prevent the animal from laying eggs. And so if you had a maternal effect lethal, the animal would make its eggs, but since the mother put in a wild-type copy of the gene, then these eggs would hatch and kill the parent, so then you'd have the progeny and the expected ratios. When the plus and M over plus guys grew up, they did the same thing. The mother would die and the embryos would hatch. But if you were homozygous for a maternal effect lethal mutation, you'd make the eggs, and the eggs would die and the mom would live. And so all you really had to do is in mass played out mutagenized F1s and then come back after a couple days and look for plates where you saw lots of larvae against and a small number of animals that had dead eggs. Pick these dead eggs and score them under a microscope. Now, when we were doing this, there were very few markers, and so it was very difficult to figure out what was an interesting gene or not. But the people then mainly involved in this are I started this work as a postdoc and then in my own lab with these are the principal screeners, and in particular Bruce Draper, Bruce Bowerman, and Craig Mello. Sometimes I refer to us as Craig Mello's first band. But they came to the lab at a time when it wasn't clear this was going anywhere. So, geez, I don't know what I'm having such a problem. They're all still alive. Anyway, Ken Kemp was an independent long-term collaborator on the project. And so the genes that we thought had clear reproducible phenotypes are listed here, and these are the dates where we published the initial gene identification, or in some cases, the mutant identification. And you can see this went over a long period of time because back then it was very difficult to clone genes. And so I'm just going to go through quickly some vignettes about what we learned. So one thing that we expected to see is no cell interactions. And this turned out to be wrong pretty quickly. So as a postdoc, I showed that in a 4-cell embryo that these two sister cells were actually born equivalent. I could show that by manipulating the positions of the embryo. And then the first mutant that I found was GLP1, which turned out to be Notch. And what it showed was that there was and then further work, that there was a Notch interaction at the 12-cell stage. And this is a ligand-expressing cell, and it induces this cell here to make pharynx. And multiple people in multiple labs then contribute to this overall picture, which was in a lineage diagram. Here's these cells that all express the GLP-1 receptor, and then there's a ligand-expressing cell that signals to these two cells here, such that these cells are induced to make pharynx, a type of mesodermal tissue. And so there's a paradox here, because these two are the sister cells, which I said are born equivalent, and yet this signaling cell, which is this orange cell here, clearly contacts these cells and induces them to make pharynx, but it also contacts these cells here, and they do not make pharynx. And so this was a mystery until we, and so then of our mutants, then these all, the GLP, and then, so we find Notch, and then we find novel components of the Notch pathway. All these mutants lack the induced pharynx. And the first big insight here was our identification of Apex1, which turns out to be delta. And this has the opposite phenotype. So instead of making no induced pharynx, you make too much. And what turns out to be going on there is that there's actually a first Notch interaction that occurs at the 4-cell stage, and it makes this sister here refractive to the subsequent Notch interaction. The reason this was missed initially is because you could go and take the ligand-expressing cell and remove it experimentally, but there was a little remnant behind, and this little remnant, because all these are maternally expressed genes, it was sufficient to signal this cell, such that the orange blastomer would only signal this red cell and not that one. And we showed that this was the case because if we manipulate the embryos but don't try to remove the blastomeres and physically separate the red one from the green one, now both of these undergo the hyperinduction. And so in terms of the lineage diagram, what we found many years later is what's going on is that there's two functionally redundant T-box transcription factors that are expressed in all of these cells here, independent of Notch. Their expression, they would have been expressed in these related cells here, except the purpose of the first Notch interaction is to prevent their expression. And in fact, if you remove genetically these T-box transcription factors, there's no necessity of this first Notch interaction. The development of this cell looks perfectly normal. And so, and in also additional studies, we show that the direct target of Notch is expressed in both of these interactions the same. And so really the difference is this combinatorial factor, the T-box transcription factors. Okay, so, and then the situation, there was even more cell interactions with Bob Goldstein's discovery that the gut, one of the earlier examples of non-cell interaction in C. elegans, was in fact due to a cell interaction, and it involved this same green cell, which is at the 4-cell stage, is the ligand-expressing cell for Notch. And so the reason I think this cell, or this interaction was missed, because it was in previous cleavage blocks experiments, it's the same story, that these early blastomeres have maternal products and they're expressed and then they have interactions, and so it was misleading. And so from our genetics, what came out of that, and this was now our lab and Craig Mellow's lab as a collaboration, and Bruce Barrowman's lab now independently, this turned out to be a Wnt signaling pathway. And so this cell here not only expresses a ligand for Notch, it also expresses Wnt. And then later we showed, in fact, there's a family of cells that in this lineage that become Wnt signaling cells that sequentially polarize the embryo, and I'm going to come back to that a little bit later. Okay, so instead of no cell-to-cell interactions, we have many rapid cell-cell interactions in C. elegans. We had hoped very much to find maternal cell fate determinants, and the big hit there was SCIN1, which is essential for the mesoderm endoderm fate that this cell makes here. And the slightly after Bruce Bowerman's work in the lab showed that SCIN1, we found mutations in genes we called PI1 and MEX1, where these cells make ectopic blastomeres produce the same cell types that this orange one never normally does. So you see two orange cells here, and here you see anterior cells. And in each case, SCIN1 activity is required for these ectopic cells to make mesoderm. So it begins to smell like a determinant, and then it turned out it was a transcription factor, so that was nice too. Now, from a genetic standpoint, PI1 then seems like it prevents SCIN1 activity from working in this posterior cell, or at least it prevents ectopic SCIN1 activity from working in the cell. But that's not the only thing it does, because if we make a PI1 SCIN1 double mutant, we find that this cell can't make germ cells. So even though SCIN1's out of the equation, PI1 is an essential gene for this blastomere in normal development to make germ cells. So when we localized SCIN1, it would have been nice if SCIN1 was only in the cell where it would seem to be absolutely required, but you can see it's a little more complex, that we see it not only in that cell, but we also see it in the germ cell precursor. Now, if you jump to the MEX1 mutant, as expected, you can see anterior mislocalization of SCIN1, but wild type, this transcription factor is in the germ cell precursor, and it also is in the posterior cell at the 2-cell stage, which is the lineage then that gives rise to the germ cell. So, but we know that PI1 is involved in this, and so PI1 we identified in 1992. We didn't get the cloning until 1996, and so this is how just illustrates how difficult things were to do, or our ineptitude, one of those two. At any rate, when we look at PI1 localization, we see it's restricted specifically to this germ cell precursor. And in fact, it follows the germ lineage trajectory of being in the precursor at the 1-cell stage, 2-cell 12, and so forth, until you get down to when this cell divides and gives rise to a clone of germ cells, and now you see PI1 in the nucleus of all of them. So it looked like that what's going on then is that you have the way that you create at least one of these somatic precursors is by restricting its activity in the posterior lineage and thus keeping this lineage totipotent, although at the same time these posterior cells clearly are expressing maternal, both Wnt pathway and Notch pathway, they're expressing signaling proteins that are, but the transcription factor is basically silenced here. Okay, so so we had initially hoped, my dream is that we're going to find a whole bunch of transcription factors that were localized in all of these somatic blastomer, but there's really only one other main candidate, which was later found by Craig Hunter and Cynthia Kenyon, PAL1, which is expressed in this somatic blastomer, and it's the same story, that it's expressed in the nucleus of both the somatic cell and the germ cell, but then PI1 basically is keeping it silent. And that just like we have MEX1, which is required to localize SCIN1 activity and protein to the posterior, then later on showed that MEX3 was doing the same thing for PAL1. Okay, so, then the other thing we expected to find and did find is proteins within the cell that were somehow resulting in the localization or asymmetric activation of maternal determinants. And this is the collaboration with Ken Kempfuse for the PAR genes, 1998, and also 1996 for PAR6. And independently in his own lab, he localized the PAR genes, and you can see these PAR3 and PAR2 are cortical proteins, and here's PI1 at the same 1-cell stage. And if you look at the 2-cell division, you can see the asymmetry of PI1, and at the same time, you see this asymmetry of these PAR genes. So there's a reiterative asymmetry. So how does this asymmetry set up? And then there was work for several years going on by several different labs that were implicating sperm entry position, which occurs normally in what becomes the posterior pull of the embryo. So sperm entry is important, and that actin by drug experiments and then staining experiments looked like it was somehow involved in actin. And our contribution here was that we were, when we tagged non-muscle myosin, that's this here, you can see what's going on is that you have a dynamic contraction of actin on the one-cell cortex, and then an asymmetry at the one-cell stage. This is all before the first division, and that the PAR proteins, the anterior ones, seem to ride this, this contraction to the anterior. And then in 2000, we published two partially redundant genes, MEX5 and MEX6, and found that they were cytoplasmic proteins with a reciprocal expression to pi1. And even in this next cell division, in the germ cell lineage, you can see the reciprocal expression. And in later work, we showed that MEX5 has a critical phosphorylation, and that one of the PAR genes, PAR1, was required for that phosphorylation. Okay, so from what I've told you so far, C. elegans, even though we went into this thinking C. elegans was going to be a very exceptional, very, very different embryo, this sounds like it's not that maybe highly unusual, but that would be missing the main point because of things like this. And so Solston's result was showing that there was an AP coupling between cell fates and division. How in the world does the cell know what the anterior and posterior side of the division is? And so when I was looking at this, I was interested in this cell here, which is the first cell death. It's a programmed cell death, and it occurs at a time when there's about 150 cells in the whole embryo. And so if I go in at the 2-cell stage, I kill this cell here, and then I let it divide, and I kill this one. I let this one divide and kill this one. So this is the only cell that develops in the entire embryo. What you can see, unbelievably, one of the remarkable things I've ever seen in science is you can just watch it, in this case, it's PAAPP death. And it was easy to score because the apoptotic cells you can see in the light microscope, so it was ambiguous. So now if I abstract this part of the lineage, the PAAPP, like this is the cell that does it, and it requires skin 1 activity to do it. Well, if we now go back and look at the mutants that have ectopic skin 1, so here's pi1 and here's MEX1, what we find, this is the cell that makes mesoderm, like this cell normally does, but if you look at it divide, PAAPP cell death. And if you look at a MEX1 mutant, all over the place, PAAPP cell death. And so what we said is that these ectopic lineages in these mutants suggest that there's a general AP coordinate system throughout the embryo. And so, and then thereafter, this is what happens when you mislocalize the skin 1 transcription factor. Same thing happens if you mislocalize what we now know as PAL1 with MEX3. Here's the wild type embryo, here's an anterior cell, here's what it normally does, makes a lot of pharyngeal cells. Here's a somatic cell in the posterior, it does something entirely different, it makes skin and muscles. In a MEX3 mutant, now we find these cells make skin muscle, but the important point is they're making it through the same AP pattern. So the idea is that there's an AP coordinate system, and in the presence of the appropriate transcription factor, it's being coupled to an output of specific cell fates. This is what makes C. elegans embryo, well, I think amazing, but it's also maybe different than other organisms. And what does this involve? It turns out that it involves a TCF-LEF1 transcription factor, POP1. And what happens is that here you see the embryo, there's probably about 80 cells here, at the time, is that every time cells divide AP, this transcription factor has high levels in the anterior daughter and low levels in the posterior daughter. This asymmetry kicks in by those the Wnt signaling events I was telling you happen in this early stage. First you polarize these cells, and then later, as later descendants express Wnt, then you polarize these cells here. And so you set the whole embryo up where they're just flip-flopping POP1 at each division. So a cell in yellow would have a POP1 history of of high, 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 but the neighboring cell would have low here, you can figure it out for yourself. And so the idea is that this is a combinatorial factor for a lot of different other cell transcription factors that determine cell fate. And we showed this is true for a wide variety of cells. It was independent of what the cells actually become. In each case, if we remove POP1 at appropriate time, we can see that, as you'd expect, that the fate is driven to the posterior cell fate. And that the Wnt mutants, where you have the converse, that you can get the anterior cell fates. I should say that this same conclusion was reached independently by the Schnabel lab, showing that the LIT1 kinase, though LIT1 doesn't show this kind of patterning, showed that there was also this underlying binary, if you want to call it, specification to cell fate. And this is what enables you to take like an early cell in one part of the embryo, and then where it undergoes from this point on a very complicated lineage. In the C. elegans lineage that Solston did, you can see examples where that same type of event happens in a completely different cell. It does exactly the same thing in all detail. And the general idea then is that it doesn't matter what happened up here, that once the transcription factor is coming on in this lineage here, it's coupled with POP1, so the output is the same, but you get the A fate coming from the same point in the lineage. And there's examples of that we've shown where you have exactly this in the notch pathway, where you'll have notch will turn on a direct target in this type of lineage, but there will be a notch independent event that turns on the same target in a different cell, and the output, or i.e. the lineage, is exactly the same. So so that's it. So that's basically that's what we did in the early embryo, and so that's what I've talked about here. And there are a lot of people involved and a lot of collaborators. These are the main ones. We didn't stop working on biology, but I thought that was the purpose of what I was supposed to talk about here. We've continued to work on adult germ cells, and we found some interesting things. And we've also worked on epithelial polarity and tissue morphogenesis. And in the adult germ cells, we worked on P granules, traffic to oocyte, and this was an interesting study where we found the first example of a transformation of adult germ cells into somatic cell types. So if you remove one of those RNA, it's KH domain protein MEX3 that operates in the embryo, and a second KH domain protein GOLD1, we see that these adult germ cells, neurons, muscle cells, and even gut cells, and that what's being activated are some of the very pathways that normally work in embryogenesis. And so here is a triple mutant where we've removed one of those transcriptions factors that's involved in muscle, and you can see the decrease in muscles here. And this is just a high mag showing that these neurons actually form long processes and look very much like neurons. The other things we've worked on are a germ cell retrovirus, and we're continuing to understand how that navigates germ cell biology. And I've been interested for a long time in apoptosis, since nearly half of the germ cells die. And that's led me lately to our recent publication about nuclear fat. It turns out that germ cells, in every single gonad in C. elegans, there's a set of germ cells that have got fat inside the nucleus. It's a lipid droplet. And by mutation, we can actually transform 1/3 of the volume of a germ cell nucleus into a big clump of fat. And it does not trigger apoptosis in C. elegans, but it does cause significant damage to somatic cells. And so we're still trying to that, and then the retrovirus, I think, are my most current research interests. Oh, and then I have to thank many people over the years that have given me much critical advice. So, thanks. So, I think I'm trying to skip this.
Okay, I hope you have it in you for one more. We'll try to make it quick. So I thought I would start right away because Jim has given such a good introduction, I can just start with the first experiment that I did that got me started on this germ cell journey. And this was kind of at a low point in my postdoc where my main project was not going very well. And at this point, I was just trying to look for genes with interesting expression patterns in their early embryo, and we wanted to focus on zygotically expressed genes. And I was doing in situ hybridization to pretty much any gene I could make a probe for. And here is the pattern that I kept seeing over and over again. This is a 4-cell embryo. And here you see the RNA being transcribed in these 3 nuclei, but not in this germ cell precursor. And we kept seeing this over and over again. And then eventually, a colleague at Hopkins, Jeff Corden, told me that RNA polymerase, you can tell when it's active using an antibody that recognizes the phosphorylated form of RNA polymerase. And we were able to show that indeed, the germ cell precursors do not have active RNA polymerase, whereas, of course, the somatic cells actively turn on RNA polymerase. And this was such a good antibody that it allowed us to go downstairs to Debbie Andrews' lab and get some Drosophila embryos and show the same thing. So you can see the somatic nuclei here positive for this antibody, whereas the pole cells over here are completely dark. And so this was the first realization that there was a fundamental difference in the way that somatic cells and germ cells turn on zygotic transcription. And looking back at this lineage of C. elegans, we found that all of these germline blastomeres and the early PGCs keep transcription off, okay? Whereas the somatic cells turn it on. So this was a cute observation, but without a mutant that would disrupt this phenomenon, we didn't know how important it was. And so to the rescue came these guys, Jim and his 2 postdocs and many more who were doing, as you saw, many screens for maternal effect mutations. And they identified this gene, PI1, that really caught my interest because it was expressed in the nuclei of all these transcriptionally silent germ cells. And I still remember how excited I was when Craig sent me the PI1 mutant, and I could show that now transcription is activated in that mutant. Turns out that PI1 is a general repressor of RNA polymerase II activity. Okay, and what's really nice, as Jim told you, in this PI1 mutant, this cell here is adopting the fate of a somatic cell, and you do not get any germline. So this was the first hint that to start a germline, you need to repress transcription so that this cell does not become a somatic cell. All right, so then, of course, I became interested in how is PI1 becoming localized asymmetrically. And for that, of course, we had a very beautiful setup from Susan Strom, Jim Priess, and Ken Canfuse, who had figured out the basic genetic pathway of how the embryo becomes polarized. And I just want to show you a little movie of how in just 20 minutes, this C. elegans embryo, 1-cell embryo, polarizes proteins at the cortex and in the cytoplasm, including PI1 in this nucleus and the P granules that Susan Strom had described. And so we started to ask basic question. What is it about the sperm that would polarize the egg? How do these amazing gradients in the cytoplasm form? And how do the P granules become polarized? So that first question was tackled by Matt Wallenfang, who was the first graduate student in my lab, and then later on Futi Motegi, a postdoc in my lab, who asked, how is it that the sperm polarizes the egg? And what they found is that the sperm brings a sperm aster, and it's this microtubule structure that initiates polarity. And one of the ways that it does that, you saw from Jim, is by tickling the actin cortex and causing these massive flows. But it also, in addition, the microtubules interact with this protein part 2 and gets part 2 onto the cortex. So that's how you break symmetry. So the next question is, how do you make these amazing gradients? At this time, the only way to make gradients that we knew about was from Drosophila, the bighoid gradient that was formed by a localized RNA that was translated and the protein would diffuse away. Here we knew that MEX5 was not made from a localized RNA, and in fact the MEX5 protein started out uniformly distributed. But what Eric Griffin found is that MEX5 protein diffusion was very different in the anterior cytoplasm, where it was diffusing slowly, versus the posterior cytoplasm, where it's diffusing fast. And as Jim told you, PAR1 is the kinase that phosphorylates MEX5, and we were able to show that phosphorylation causes MEX5 to move fast in this part of the cytoplasm. It's moving slow over here. And so that's how gradient forms, by local regulation of protein diffusion, or just by causing a traffic jam in the anterior cytoplasm where all the proteins are moving slowly and accumulating there. So a way to make a gradient without localizing RNA. So then we finally got to my favorite project, which was to figure out how do P granules polarize. And this is really fascinating because these granules start out uniform into the cytoplasm, and Susan Strom had already noticed that the granules in the most anterior seem to disappear and the ones in the posterior grow. And Cliff Brangren and Tony Hyman, when they made similar movies, suggested that what was happening is that these granules were droplets, and liquid droplets that would pretty much evaporate or dissolve over here and become condensed over here. Okay? And this idea really got a lot of traction when Tony Hyman's lab showed that PEGL1 and PEGL3, these proteins that are in the P granules that Susan Strom had identified, these proteins readily phase separate in vitro, meaning you can take this protein in a test tube and it will form a dense little protein droplet that looks like a little water droplet, and you can see these two fuse with one another. Okay, so that was in vitro, and of course we wanted to know, how do you go from this droplet behavior to localized dissolution and growth in separate parts of the same cytoplasm? And so this took a lot of people. I'm showing you some of them here, because we had to do genetics here to identify new components of the P granules. PEGL alone was not enough. And we first had to identify a kinase that was required to dissolve the P granules in the anterior cytoplasm. We found a phosphatase that had the opposite phenotype, where now the granules were dissolved everywhere. And then, using this phosphatase and some clever ideas that Jen Wong had, we finally got to the key proteins. And these are these intrinsically disordered proteins. These are not proteins you could have found in the genome just by looking at sequence, because they looked like nothing. They're not they're not evolutionary conserved in the sense that you can't find a homolog easily, but they are, have a structure that is similar to this family of protein that was later described by the Page lab, with a long intrinsically disordered domain and an ordered domain. Okay, so these proteins, we found them because when you get rid of them, the P granules really are very confused. And this is one of the phenotypes where in wild type you see the beautiful P granules all over the cytoplasm. This is before polarization. And here, now you see fewer granules and they are larger. And this was really odd until I started to learn some basic physical chemistry about how liquid droplets or emulsions behave. And I learned that emulsions, so collections of droplets, are inherently unstable. And that is because the system wants to minimize surface area and maximize volume. And so one way to do that is to get all of the droplets to fuse with one another, so you maximize the number of molecules that are inside the droplets, making lots of nice interactions with their neighbors and avoiding interacting with the solvent. And you might be familiar with this concept if you've ever made a vinaigrette in the in the kitchen, sorry, not in the lab, you'll know that if you mix vinegar and oil, if you shake your vinaigrette, you will get a very nice emulsion with very tiny oil droplets nicely dispersed in the vinegar. However, if you wait for a while, this is what your vinaigrette is starting to look like. You'll see all the oil droplets will fuse with one another, and because they're lighter, they will separate completely from the vinegar, okay? So this was very similar to what we were seeing in these MEG3/4 mutants, where the P granules looked like they were coarsening. This process of going from many granules to just many droplets to just a few, it's called coarsening. Okay, so we wondered, well, how do the MEG proteins prevent this P granule coarsening? Helen Schmidt realized that the MEG proteins are actually bipartite proteins. The long intrinsically disordered domain is what binds RNA. That's actually how the MEG proteins bring the RNA to the P granules. But the ordered domain has a little motif that binds to PEGL. And Andrew Folkman was able to show that MEG and PEGL come together in the granules, but in an interesting way, where PEGL is a liquid droplet, it's nice and round, but MEG makes these unusual clusters on the surface of the PEGL droplets. So this is in vivo, in a real P granule in vivo, but we also were able to reproduce this in vitro, and this is thanks to Andrea Putnam, who's a biochemist who was able to purify full-length PEGL3, full-length MEG3, mix them together in a test tube, and just spontaneously you get the PEGL3 droplet and then the beautiful MEG3 clusters on the surface, okay? Not only that, but Andrea realized that the MEG3 clusters were very different from the PEGL droplets. This is a liquid, okay? These things are very sensitive. If you add a little salt, they'll dissolve. These MEG3 clusters are solid. They resist dilution. You can add salt. They are like little rocks, and they are absorbed to the surface of this liquid droplet. All right, so that was the conclusion. We had solid MEG3 clusters absorbed onto these liquid PEGL3 droplets. Now, we were kind of stuck for a while here until I finally, out of desperation, literally Googled solids on liquids and how would you stabilize an emulsion with having a solid on a droplet. And this is where Google is awesome because Wikipedia immediately gives you the answer. This is a very well-known phenomenon from physical chemistry. Pickering emulsions have been known by chemists for over 100 years ago. They were discovered by Spencer Pickering and Ramsey. They were trying to stabilize basically vinaigrettes, oil in water emulsions, and they realized if they added clay, they could stabilize the droplets because the clay formed little aggregates that would go on the surface of the droplets and prevent the droplets from coalescing with each other. You might think this is very esoteric, but you probably have yourself made a Pickering emulsion in the kitchen. And that is, if you want to prevent your vinaigrette from separating, one secret that French cooks use all the time is to add mustard. And that is because mustard seeds, the shell of the mustard seeds, have tiny little particulates that will sit on top of your oil droplets and prevent them from coalescing. And so you can stabilize your vinaigrette at least until your guests arrive. Probably not until tomorrow, but for a little while. Okay, so then we asked whether our MEG proteins would do this. So Pickering agents have been very well studied in physical chemistry. This is a big deal in the food industry, the pharmaceutical industry, but they had never been described inside of cells. And so we asked whether our MEG proteins could function like Pickering agents, and this is one of the experiments that Andrea and Andrew did, where you can take a PEGL3 emulsion and let it sit over time, and you'll see it coarsen with just fewer droplets that are larger. But if you add MEG3, now you can stabilize the emulsion for the duration of the experiment. And that is the signature of a Pickering agent. And that's exactly what MEG3 does in vivo. Because when you have MEG3, you see the nice dispersed peggle emulsion, and when you don't, the pegels start to fuse with one another. Another thing that Andrew realized is that during polarization of the zygote, MEG3 is one of these proteins that becomes localized in a gradient. And so the peggle granules all start out with a little MEG3 coat, but only the anterior ones lose their coat, only the posterior ones have their coat. And so that stabilizes the P granules in the posterior side of the cytoplasm, and then causes these guys to become unstable and lose their peggle material to the posterior ones. And so there you go. We now understand how P granules become asymmetric. It's due to a polarized Pickering agent that stabilizes the peggle droplets. This is nice cell biology. It tells you how things move around. Now we want to know, what do they do? Remember, we're still in the business of understanding how you make germ cells. And the next part is going to be to try to understand what is the function of these P granules. So there we got inspiration, of course, from the work of Ruth Lehmann in Drosophila. And we already knew about nanos RNA being an RNA that would be in the germ granules of Drosophila and important for germline development in Drosophila. And so Subu is the first postdoc in my lab, he wanted to look at nanos homologs in C. elegans, and indeed found that this nanos homolog localizes to the P granules and segregates to the germline founder cell, and that it's where it's translated. And Subu was able to show that when you knock out nanos, you do not make germ cells anymore, and the animals are sterile. Okay, so next, we wanted to know, well, is it important that nanos is in germ granules? And this is something that you've heard about just a couple days ago from Alicia, where if we now take our MEG mutants where the germ granules are not segregated asymmetrically anymore, we see that nanos is not in germ granules, and however, it's still stabilized in the germline founder cell. It's not enriched anymore because it wasn't in the granules, but remarkably, it's still translated on time, and these animals are mostly fertile. So again, we were stuck here for a little bit because we really thought the germ granules would be regulating the RNA translation, and until Madeline Cassani, who again you heard about a couple days ago. When Madeline came along, she was interested in these other intrinsically disordered proteins that resemble MEG3/4, but they don't have this peggle motif. And what Maddie was able to show is that these form separate bodies, the germline P bodies, as we call them, that can associate with P granules, but also exist all throughout the cytoplasm. And those are the ones that are regulating nanos translation. When we get rid of these guys, nanos is still in the P granules, and it still is enriched in the germline founder cell, but now it stays in the granules, which is remarkably like what you heard Erez Raz talk about when nanos could not get out of the zebrafish germ granules when DND1 was not around, okay? So that's the same thing. The nanos RNA in this situation stays in the granules, and it's not translated efficiently, and these guys are 100% sterile. Okay. So the other interesting thing that Maddie figured out is these mutants are sterile because now P4 is adopting a fate that's very similar to D, the muscle precursor, okay? And they go on to express transcription factors typical of somatic cells. So we also found out that when you get rid of nanos, I told you that you're also can't make functional germ cells. And Sean Lee in the lab did transcriptomics on these germ cells and figured out that what's wrong in the nanos mutants is that there's a profound failure to degrade many maternal RNAs, some of which codes for somatic transcription factors, and so that's why these cells are confused and cannot become germ cells. So to put all of this together, there's really 3 different independent ways that these germ cells are trying desperately not to become somatic cells. First, using Pi1 to just globally stop transcription. And this is very important because otherwise you lose your germ cells very early in this lineage. Next, with the germline P bodies to prevent P4 from becoming D. And then later with nanos to prevent these guys from becoming confused. Okay, so several layers of preventing somatic development. So from this, we really think that even in C. elegans, where you might have thought that the germ cells were very close to the oocyte and might have been germ cells all along, what we really find is that these cells are derived from these precursors that are mesoderm fated, and you need to downregulate this inherited somatic program. And I would like to venture to say that this is very similar to what we see in other embryos, such as mouse and human embryos, where the PGCs are also derived from somatic cells, from mesoderm-fated progenitors that need to downregulate the mesoderm program, turn on nanos, and become PGCs. And so you'll recall that early days of this meeting, we used to make a big deal about the difference between animals with germplasm, such as our favorite model system, that would have preformation, where the germ cells were always germ cells, versus epigenesis model, where the germ cells need to be induced. And this is actually the mode that is the most common in evolution. And we would argue that these two are actually very similar mechanisms. The only difference is where do you get your start? Is it from signaling cues that cause the downregulation of these mesoderm transcription factors and activation of nanos, or is it using these maternal germline P bodies that again downregulate somatic factors, activate the translation of nanos, and set you on your way to make germ cells? And so this is where we're going to go in the future, is to really study how do you downregulate this mesodermal fate to allow for the germline fate to take hold. So this is all I wanted to tell you. These are photos of my lab over time. It has evolved and changed. We have had some really fun times together, some more difficult times as well. But throughout, it's been a wonderful ride, and I am very grateful to my lab, to the Gruber Foundation, and to all of you for your attention. Thank you.
This concludes the Gruber program. I know we're holding you back from the drinks reception, so we'll see you there, and congratulations again to our 3 recipients.